All right, well, as we continue our crash course through molecular genetics, we're going to talk about replication. So here are the key terms that we're going to discuss through this presentation and also in our class on Wednesday. First one, polymerization, the process through condensation or dehydration reactions of adding monomers into or stringing monomers together to form polymers. DNA polymerase is an enzyme that attaches DNA nucleotides to an already present DNA nucleotide. It is, except for one specific type in eukaryotes, it is not self-priming, meaning it cannot add the first nucleotide, but rather only add new nucleotides. Helicase is an enzyme that breaks the hydrogen bonds holding the two strands together, and so sometimes it is referred to as the zipper, unzipping the two strands apart. Single-stranded binding proteins. These are proteins that prevent the two strands from recombining before the machinery has a chance to get access to the strand to read it, to write the new strand. Primase is the enzyme that is self-priming. It writes actually an RNA primer to begin the process of DNA polymerization. That primer will then need to be replaced later in the process by DNA nucleotides. Ligase is an enzyme that fixes nicks. You'll see as we talk about this process of replication that there are a lot of nicks that end up appearing inside of typically your new strands during DNA replication. Okazaki fragments, these are short sections of DNA that are written on what's called the lagging strand. So as DNA polymerase works, not only is it not self-priming, but DNA polymerase can also only write in the 5' to 3' direction, meaning it can't add the first nucleotide, and it can only attach a new nucleotide to a free carbon 3 on that deoxyribose sugar. And so because of that, there's one strand that cannot be written continuously. It has to be written discontinuously in short fragments called Okazaki fragments. Topoisomerase or DNA gyrase, two names for the same enzyme, helps to decondense the DNA to make it easier for the machinery to gain access to these strands of DNA. All right, so right at the time that Watson and Crick proposed their model for DNA as a double helix, they also presented a mechanism by which it could be replicated, and that was the double helix itself. And so as you look here, we've got these antiparallel strands, this one running 5' to 3', this one running 3' to 5' or 5' to 3' in the opposite direction. So we already talked about these bases bonding together in very specific pairings. And so as you are adding new nucleotides, DNA polymerase can read what's here on this template strand and attach the only nucleotide that has a base that will actually form hydrogen bonds with this. And the beauty of it is basically there's two options. You either form hydrogen bonds or you force these things too close together and they won't hydrogen bond, and that's because basically the same charges are bumping up against each other. You have to remember that hydrogen bonds form between polar molecules. So this base right here is polar, this base right here is polar, and so a hydrogen bond will form when the positive pole of one interacts with the negative pole of another, each of these hydrogen bonds. So if they can't hydrogen bond, you actually have the positive pole of one being forced to interact with the positive pole of the other, and they actually repel each other, and it creates a bubble if the wrong nucleotide is added. Another thing to point out is when these nucleotides come in, they are in the form of nucleoside triphosphates. And so in this case, this is would be it'd be tyrosine triphosphate, and in order for it to be attached to this carbon 3, we've got to form a bond between this phosphorus this phosphorus right here and this oxygen right here. And so in order to do that, we actually have to remove two phosphate groups. And we see when you take a phosphate group off of ATP, it releases energy. Same thing happens here. And so these nucleotides bring in the components necessary to actually facilitate this process, to fuel the process. And so as we continue to talk about this, we need to talk about not just how do we add new nucleotides, but how does this whole process work. And so here, this is in prokaryotic cells. We've got these repeats upstream from where the replication actually begins. And so we've got AT-rich 9 base pair repeats and 13 base pair repeats. And the significance of that, if we go back here, you'll be reminded that A and T form 2 hydrogen bonds versus the 3 formed between guanine and cytosine. And so in an AT-rich section, you're going to have the strands less tightly bound together because you're going to have only 67% of the number of bonds that you would have in a GC-rich section. And so you've got a protein called DNA-A that will come in and will denature these two strands. Once they are denatured, helicase will come in, and remember, helicase, again, it breaks double bonds, and so it moves in both directions, essentially unzipping these two strands from each other. And so now we have space where we can actually work. We can bring in bulkier machinery. Here's DNA primase physically attaches to the helicase, and so as helicase proceeds in this direction, the strand it's reading is in the 5' to 3' direction. We know that primase and DNA polymerase can only write in the 5' to 3'. So if it's reading this way, it would have to write in the 3' to 5'. That's noted here. This is the 3' end. This is a 5' end. It cannot write in this direction, so it would only be able to write in this direction here. Single-stranded binding proteins are going to come in and keep these strands from reattaching. And as we look here, we'll focus in on one side of this. This right here is called the replication bubble. So we'll look over at one side. Here you would have helicase breaking these hydrogen bonds, unzipping these strands. And so that's going to be happening here. It'll also be happening on the other side of the bubble, but we're just focusing here. So on this strand here, this would be the continuous strand. The primase was able to come here. It read this in the 3' to 5'. It wrote 5' to 3', which is the only direction it can write. The reason why primase comes in, again, is because DNA polymerase is not self-priming. It cannot add wanted to make sure it was recording. It cannot add the initial nucleotide. So primase will come in, write a short RNA primer, DNA polymerase will come in and take over, and it will just keep writing all the way until it gets to wherever the end is. Now, in this, we're looking at a bacterial chromosome, which is going to be circular, and so it's just going to write all the way around until it comes back to where it started. Now, on this strand, if it was going to move in this direction, which helicase is moving in this direction, primase is physically attached to it, would be moving in this direction, but it cannot write in that direction. So instead, what it has to do is when it gains access, enough room, it will attach, it will write a short primer in the opposite direction. DNA polymerase will take over and will keep writing until it reaches another one of those RNA primers. And then it'll fall off, and you'll have to have a different DNA polymerase that can come in that can write over the RNA nucleotides. but in the interim, you end up with these Okazaki fragments. Again, these short pieces of DNA, and then a new DNA polymerase will come in, will replace all of these RNA nucleotides with DNA nucleotides, but we'll be left with, so here's DNA polymerase 3 that comes in, that writes over here, and then you have DNA polymerase 1 that will come in, that will write and replace all of those RNA nucleotides with DNA nucleotides. But again, it cannot create a bond with carbon 5, which is what it's going to run up against here. It's going to end up with a nucleotide right here. Let me get my pen. It's going to end up with a nucleotide right here that has a free carbon 5. But it can't create that bond there. It can only create a bond where you've got a free carbon 3. So you actually need another enzyme called DNA ligase to come in and create that bond between the carbon 3 from the last nucleotide, DNA polymerase 1 added, and the carbon 5 of the first DNA polymerase that was there following replacing the RNA nucleotides. So in a way, this DNA ligase does what we call gluing these Okazaki fragments together. Now, a little bit more about DNA polymerase 1. DNA polymerase 1 is the bacterial polymerase that switches the RNA nucleotides for DNA nucleotides. And so it's obviously involved in writing strands of DNA. It, like DNA polymerase 3, has what's called the 3' to 5'. Notice that that's in the opposite direction of which they write, but 3' to 5' exonuclease activity. Exonuclease means it can remove nucleotides. And so, like DNA polymerase 3, DNA polymerase 1 can back up and pull out a nucleotide if it added the wrong one. And again, as I mentioned in the previous slide, it'll know that it added the wrong one because instead of forming hydrogen bonds, you'll get some repulsion and you'll form like a bubble or a road bump. But unlike DNA polymerase 3, DNA polymerase 1 also has this 5' to 3' where it can cut out as it's moving forward. This allows it to remove the primer, those RNA nucleotides written by primase to start the process, and replace those with DNA nucleotides, deoxyribose nucleotides. Now, when you study this protein, you find out that there are actually two main domains. There's this domain here called the clinal fragment that is responsible for writing the strands in the 5' to 3', and also for this 3' to 5' exonuclease. These two functions here are carried out by the clinal fragment, and the third function here is carried out by this part that is cut off to form the clinal fragment. And the significance here is this fragment here, the clinal fragment, is incredibly efficient and is actually a wonderful tool that we've been able to use for in industry for actually writing DNA strands, for sequencing reactions, for writing strands of DNA that people order. And so that's one that we'll actually talk a lot about in this class as our class goes on. All right, so in prokaryotes, we have at least 5 DNA polymerases. We've got the main DNA polymerase, which is DNA polymerase 3. This has the 3' to 5' exonuclease, that proofreading where it can back up and remove incorrect nucleotides. And then it is the main one in that it is the one that writes most of the new strand. It adds most of the new nucleotides. But DNA polymerase 1 is the one that is necessary for replacing the RNA nucleotides with the with DNA nucleotides, and this is because it's got this 5' to 3' exonuclease activity. Notice DNA polymerase 2 also has this 5' to 3', 3' to 5' to 3' extension ability. It does not have the 5' to 3' exonuclease like DNA polymerase 1 did. So it's involved in replicating damaged DNA. These DNA polymerases 1 and 3 will typically, if it gets to a section of DNA that is damaged, that the wrong base is there or things aren't attached correctly, they will not write a new strand. And so in some sections of the genome, they still need to be written even if there are small errors, and DNA polymerase 2 will take over that. DNA polymerase 4 and 5 will also function to write damaged DNA. All right. So when we're talking about replicating an entire bacterial genome, at least as far as we can tell from E. coli, but is probably the case for most bacteria, and for writing really large plasmids, this is the mechanism of DNA replication we get. It's called bidirectional replication. So here you'd have like that 13 base pair repeat and the 9 base pair repeat, those AT-rich sections. You would get DNA A that would come in and denature them or separate them. Locally, you'd get helicase that would come in and would unzip it in both directions. You would get primase attached to those helicase that would write 5' to 3' on the leading strand, would also write 5' to 3' on the lagging strand, but as it was able to gain access, you'd get those Okazaki fragments. And so this replication bubble will grow, moving in both directions, creating what's called the theta structure, which is a Greek letter that looks just like this. And it will proceed all the way around until you replicate both, or you use both old strands as templates and write a new strand with those old strands. And then you get to the end, and there's another enzyme that will separate the two from each other. And now you've got two new entire bacterial genomes or large plasmids. And notice that both of them are what are called semi-conservative, in that they both have one old strand and one new strand, and they both required writing in both the writing at each side of the replication bubble, on both the leading where it went all the way through and on the lagging strands. All right, so another form of replication is rolling circle replication. This happens in some bacterial plasmids and in bacteriophages, viruses, and probably many eukaryotic phages as well, although those tend to have both bacteriophages and eukaryotic viruses tend to have single-stranded DNA, but this is called rolling circle replication. And so what will happen here is you'll have an enzyme that will make a nick here that will separate one, will create a separation between two nucleotides in one of the strands, and then this will open it up for helicase and primase and DNA polymerase to gain access, and it will begin writing using one of these things, using the non-nicked strand as the template strand. And as it does, it will displace this nicked portion off to the side, and then it will just keep replicating all the way around, displacing that other ring more and more and more until it gets all the way around and it's read the entire template strand, written the entire new strand. Now, at the same time, you can have these Okazaki fragments being written on this displaced strand, so that you're actually still getting, when you're at the end, two entire chromosomes, one with, or two entire plasmids, one with an old strand, or sorry, both with one old strand and one new strand. Now, while this is happening, you can just have many cycles of rolling circle replication, where you just continue to displace one of the strands as basically this machinery just keeps wrapping and wrapping around that circle using one of those strands as a template, and it can just keep reading the same one. So after it gets all the way around here, you can have that enzyme come back, create that same nick that was created here again, and then just write, now displacing that new strand you just wrote, while again you can have machinery writing Okazaki fragments on that displaced strand, and you can just keep doing this, replicating this plasmid over and over and over again. And these are called concatamers, when you're forming basically strand after strand after strand after strand, and oftentimes it's happening so rapidly that while you're only halfway done, say if we go back over to here and try to erase a little bit of this, and so say you got the machinery in here, and while this was only halfway done, that machinery came in, created the same nick, but now here, and another one starts writing while this one's only halfway done. And so then you can get this really complex structure where you've got this chromosome with several rounds of rolling circle replication happening at the same time. All right. Eukaryotic DNA replication is very similar to prokaryotic DNA replication with a couple of exceptions. Eukaryotic DNA replication, when it happens, is very different than prokaryotic. Prokaryotic cells replicate so quickly that the DNA, the main bacterial chromosome, is almost constantly being replicated. On top of that, bacterial cells exchange plasmids so readily that those plasmids are basically always being replicated. Eukaryotic cells only replicate their DNA during S phase of the cell cycle, and only when you have two things that happen. One is you have to form what are called the pre-replicative complexes, or PRC complexes, during G1, during growth 1. You have to form these complexes that will trigger the replication events in the presence of certain kinases during S phase. So you've got a couple of cyclin-dependent kinases here. Here's cyclin-dependent kinase 2, cyclin-dependent kinase 2. When these kinases activate, if you will, these cyclin A and cyclin E, and you've already formed that pre-replicative complex, or the PRC complex, then finally the cell can finish S phase by duplicating all of the chromosomes within the cell. So that's one big difference in eukaryotic replication. The other big difference is, rather than having a single circular chromosome that tends to be relatively small, you have several linear chromosomes that can be incredibly large. And so this morning I told you that I was thinking that chromosome 22 was 4 to 8 million base pairs in length. It's actually 49 million base pairs in length, which in terms of the number of bases makes it the second smallest chromosome. And so you can just imagine those are a lot of bases that need to be written during this process. So another difference is the number of DNA polymerases. So in eukaryotic cells, we have at least 15 DNA polymerases, many of which we don't know what they do yet. So DNA polymerase delta is the equivalent of DNA polymerase 3 in prokaryotes. It is the main DNA polymerase. It has this 3' to 5' exonuclease activity, and writes the most nucleotides on those new strands. DNA polymerase alpha is equivalent to primase, writing that RNA primer, but then it also is actually equivalent to DNA, maybe to DNA polymerase 1 in bacterial cells, in that you have it kind of writing a small number of the bases. DNA polymerase epsilon does actually work a lot more on the lagging strand, which is where you see DNA polymerase 1 acting a great deal in prokaryotic cells. And then we've got DNA polymerase gamma that replicates mitochondrial DNA, DNA polymerase beta that actually works towards repairing DNA, and then you've got all of these polymerases that, like DNA polymerases 4, 5, and 2 in prokaryotic cells, that can bypass DNA damage. And then we have several others like theta, lambda, phi, sigma, and mu that we don't know exactly what they do or where they function entirely as of yet. Another big difference between eukaryotic and prokaryotic DNA is that because of how large these chromosomes are, you don't have a single origin of replication like you do in bacterial DNA replication, whether you're doing rolling circle or bidirectional replication. You have a single origin. And on eukaryotic chromosomes, you have several origins of replication. The larger the chromosome is, the more origins of replication you have. And then so DNA polymerization will continue all the way up until the point where it bumps up against another place where you've already had primers, or where you've already had primers, already replaced those primers, and you'll get a nick because DNA polymerase can't create that bond in there, and you'll get something equivalent to ligase that will come in and will fix those nicks. So those are the main differences. And then we need to talk a little bit about telomeres. And this is really what we're going to focus a lot of our time on Wednesday. We'll talk about the replication bubbles, the leading strand, the lagging strand, get all that sorted out if it wasn't clear in this video. And we're going to focus a lot on telomeres. Now, you'll notice from looking at this, based on what we were talking about in our lecture for Monday, is that this chromosome isn't drawn exactly right. This 3' end should extend out beyond the 5' end. And so here you've got our replication bubble. Here you have the leading strand where DNA polymerase is able to write continuously in the 5' to 3' direction as it reads 3' to 5' on the template. Here, the DNA polymerase has to write in this direction because that's the only way it can write 5' to 3', because it cannot write in the other direction. And so we're coming in here, we're making these Okazaki fragments. We've got, DNA polymerase alpha coming in eukaryotic cells and writing these primers, and then giving way to DNA polymerase delta that writes more all the way up until it replaces those primers and creates a nick, and then something will come in and fix those nicks. But this will continue all the way on this leading strand here until it gets to the end of this 3' strand. It will write all the way until the end there. But now this one, as it writes, it will get to a point where this bubble will keep opening up and up and up. Let me erase what I wrote here so I can draw something a little bit different here. This will keep opening up. This will keep opening up. These strands will keep getting further apart, and you will keep writing these short Okazaki fragments here, but it'll get to a point where there is not enough room. Here, let me still extend this out the way it's supposed to be. It will get to a point where there's basically not enough room for DNA polymerase alpha to get in to read this template strand here and to write an RNA primer that's complementary to it. And so every time we replicate this, one of our strands was able to write all the way to the end here. We were able to, on this leading strand, get all the way to the end. And so that version of the chromosome was actually able to write it actually even becomes longer than what the original one was, because now this strand, instead of starting here, would actually start all the way back here as it read this template strand and kept writing. But this one here, actually, this one on this side, on the top here, actually ends up shorter than what the original chromosome was, because it gets to this point where you can't get, again, it can't get DNA polymerase alpha in here to actually write that RNA primer. And so every time you replicate, you end up shortening one of those two chromosomes on this side, and then on the other side, the other one would end up being short. So this one would end up getting shortened, as you can't get in, DNA polymerase alpha can't get in there and write that RNA polymerase. And so you can see that illustrated here. This primer either can't get in to even write the primer, or their DNA polymerase, it'll cut that primer out, but it actually can't write anything there because there's nothing to attach it to. There's no free carbon 3, and so you end up with these holes on these telomeres. And then so you have to have a machine that can come in and will restore some of that lost information. But it can't restore all of that lost information, but it can restore some of that lost information. And so notice which one we actually shorten on one of those. So we're shortening here as we copy one of those, and this is where we're supposed to actually have a longer 3' tail. And that's what actually gets shortened on one of our chromosomes. So we will have an enzyme called telomerase that will come in. It will physically attach itself. Let me erase this, give myself some space. You'll have telomerase that will come in, that will recognize this section here, and it has a corresponding RNA sequence that will bind to here and also create a longer little tail there, that then DNA polymerase can come in and can actually write the nucleotides on this strand that are complementary to this little RNA section. Telomerase will then fall off, and then now DNA polymerase can come in and replace that with the complementary nucleotides that should be there. And so telomerase does help to prevent these telomeres from getting incredibly short, incredibly fast, but it can't eliminate all of that loss of information. That's one of the reasons why these chromosomes end in telomeres, is so that you have some space on the chromosome to lose until you start getting into protein coding regions or regulatory regions that have essential information for the life of the organism, for the life of the cell. And so, again, as you prepare for class on Wednesday, interact with the quiz, and also be prepared to talk about the leading strand, the lagging strand, these Okazaki fragments. Why is that necessary to replicate that way? The different DNA polymerases and these issues that happened at the end of chromosomes and how cells can try to prevent it. So that's it.